Hello, everyone, and welcome to today's webinar, Companion Diagnostic Approaches in the Development of Cell and Gene Therapies. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by LabCorp. To learn more, please visit labcorp.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome Akanksha Gupta, PhD, Executive Director, Cell and Gene Therapy at LabCorp. For a complete biography on our speakers, please click on the Presenter tab. Akanksha, I'll pass it over to you to get us started.
Thank you, Kayla. First of all, I would like to welcome our audience and thank the panelists for joining the discussion today. As Kayla mentioned, the focus of today's webinar is on companion diagnostic approaches in the development of cell and gene therapies. Now, cell and gene therapies are really transforming how we treat disease, offering options not only to treat, but also provide cure for the treatment of wide variety of diseases ranging from rare genetic disorders to even cancers. And companion diagnostics can play a critical role in the development of these therapies, ensuring that the right patient receives the right treatment and that the treatment efficacy is achieved, which is not only aligned with the patient's genetic as well as the cellular profiles. We're really seeing the field evolve, and we've seen some major advancements in the companion diagnostic field as it relates to cell and gene therapies. This year, we saw the first FDA-approved companion diagnostic for a gene therapy-based asset. This test, dubbed AV5 Detect CDX, is a companion diagnostic test that can aid in the identification of patients that would receive BioMarin's gene therapy drug Roctavian for the treatment of severe hemophilia. We also saw a breakthrough designation for another gene therapy asset for Sarepta for their Alvedis asset for the treatment of Duchenne's muscular dystrophy. So clearly the field is evolving, and this integration of companion diagnostics with cell and gene therapy development, it's no longer theoretical, right? We are actively seeing these strategies being pursued in the development of these cell and gene therapies. It's really causing a paradigm shift, right? It's no longer a one-size-fits-all approach. It's a personalized approach. So in today's discussion, we're going to hear from our panelists their views on the current landscape of companion diagnostics as it relates to cell and gene therapies, the different types of assays and assessments that can be utilized for the development of these companion diagnostics, regulatory strategy, as well as commercialization considerations. So without further ado, I'm gonna ask the panelists to introduce themselves, and then we'll delve right into the discussion. So Taylor, let's start with introductions from your end first, please.
Perfect, thanks Akanksha. Good morning, good afternoon, everyone. My name is Taylor Jensen. I'm a molecular biologist by training and serve as Vice President and Head of Science for Oncology at LabCorp, where our team really focuses on the development and implementation of new technologies, new assays, new tests, with a particular focus in the area of oncology, as well as companion diagnostics across all therapeutic areas, including, obviously, today's topic in the cell and gene therapy space.
Hi, this is Chris Petropoulos, and I serve as VP of R&D at LabCorp Monogram Biosciences, which is a specialty laboratory with LabCorp of America. And I've been with the company for 27 years. My formal training is molecular biology, molecular and cell biology. And my career has been focused on the development of assays to assess viral resistance and neutralization. And we specifically at Monogram engineer viruses to develop assays to address these applications. For the last 10 years at Monogram, we've actually been working on the development of assays to assess neutralizing antibody responses against adeno-associated virus. And as we'll see, that's a key virus that's used to mediate or direct gene therapies. Irene?
Hi, everyone. Irene Bacalocostantis. I am a biomedical engineer by trade and a regulatory SME. I started my career in regulatory at the FDA over 10 years ago, and I'm currently Executive Director for Regulatory Affairs Companion Diagnostics at LabCorp. Our team supports CDX development here and getting products through commercialization across different platforms, including in the CGT space. And I'm really excited to be here to talk to you about the evolution in the regulatory landscape that we've seen over the last few years.
Thank you, Irene. Thank you, Chris and Taylor, for the very nice introductions. Let's get into the discussion, right? So cell and gene therapies, right? They're frequently discussed in tandem, but they really are distinct entities, right? They have unique characteristics as well as applications, right? And this would also be applicable to a companion diagnostic that gets developed for a cell therapy versus the gene therapy based asset. So can the panel share their views on the current landscape of companion diagnostics in the context of cell therapies versus gene therapies? And Taylor, I'm looking at you. Share your views from a cell therapy perspective, and then Chris, would love to hear your thoughts from a gene therapy perspective as well.
Yeah, absolutely. Thanks, Akanksha. And yeah, like you said, I'll talk a bit more about the adoptive cell therapies, and then I'd love to hear more from Chris around what he's seeing in the gene therapy space. But when it comes to kind of the adoptive cell therapies, like CAR T therapies, I think probably many of the people on this call today are aware that there are currently 6 cell therapies that have been approved worldwide. And as it stands today, none of those have companion diagnostics linked to their prescription or use. And I mean, that's likely due to multiple factors. First, all adoptive cell therapies to date have been autologous, meaning that they've involved collecting and subsequently modifying and culturing the cells from the exact same patient that they will ultimately be transplanted back into. And so this limits the need for potential companion diagnostics today. But, as we move forward towards, a field where that may not always be the case as we transition from the autologous to the allogeneic type of therapies, that could very likely change. And it's not to say that there are not currently tests should be performed in conjunction with these types of therapies. For example, it's been well established at this point that there is a link between persistence of the CAR T therapy and outcomes of patients. And so there's been a number of studies that have looked at different approaches that we'll talk a little bit more about later, that enable the continuous monitoring to try to determine whether or not a patient is most likely continuing to respond, or if alternative therapies may be something that should be considered. But Chris, I know that there's some similarities certainly in the cell therapy space, but would certainly love to hear your thoughts on how you're seeing things play out in the gene therapy space.
Sure. Thanks, Taylor. Yeah, so I'll just start by saying that most gene therapies leverage viral vectors for gene delivery. These might include adenoviruses, adeno-associated viruses, or lentiviruses, other retroviruses. Adeno-associated virus is really the preferred approach, often referred to as AAV, just to condense the word. AAV is considered safe and effective and non-pathogenic at this point, although it is still early days. It also has the advantages that that it is able to infect a wide variety of cell types. And so it can target many different tissues in the body. As far as we know, AAV vectors stably deliver genes, although it's still early days. So if that holds up, then they provide an opportunity for lifelong cures. And so we're at a stage now where we're beginning to collect that longitudinal data. Unfortunately, AAV infection is very common in humans, and to the extent that 50% of the population may have had a previous AAV infection. So why is this important? Because when you have a viral infection, you typically mount antiviral antibody responses. And so those antibody responses, if they are directed to AAV, can limit the effective transduction of target cells and thereby limit the effectiveness of the therapy itself. It's very important, especially at these early days when we're trying to generate clinical data, to identify the appropriate study subject population, eventually the clinical patient population, identify those individuals that are AAV negative or seronegative. And there's a couple of different ways to do that. I'm most familiar with and have most experience using cell-based infectivity assays. Those are often referred to as neutralizing antibody assays or transduction inhibition assays. They're very comprehensive because they can measure any step that interferes with gene delivery to the cell. But they are of much higher complexity than some of the other approaches. They also are associated with higher costs. And it's a little more challenging to think about distribution of those wide global distribution of those assays. The other approach is just looking at binding antibodies or total antibody assays. So these are probably more familiar to a lot of people, where you're looking at some type of ELISA or antibody capture assay. This only measures antibody binding, and it really doesn't tell you whether or not those antibodies interfere with gene delivery. Oftentimes there's a very strong correlation between neutralizing antibody responses and total antibody responses, but we don't know the complete picture yet. These assays are of lower complexity and thus lower costs, and they're easier to distribute. At this point, really, there's no standard off-the-shelf assay. So we're often asked, don't you have an off-the-shelf assay? And the answer to that is no. And there are no industry standards, although some common reagents are commercially available, and that always helps with assay development. At this point, regulatory guidance is limited. There are no FDA points to consider at this point, but the regulatory agencies are becoming increasingly more engaged, and I have to say that both the regulatory agencies and the sponsors and the diagnostic companies, we're all learning as we go. It's that classic, we're building the jetliner as we taxi down the runway. To date, gene therapies have focused on rare diseases. Consequently, the trials are very small, and there's minimal clinical outcomes data to help us establish clinical cutoffs. So we'll talk about this a little bit more later. So it's very important for us now at this stage to understand how the NAB and the TAB assays impact treatment efficacy. Back to the regulatory landscape, it's evolving, it's dynamic. Regulatory scrutiny is increasing, but somewhat unpredictable. And there have been therapies that have been approved without a CDX, although Akanksha has mentioned some recent developments there. And before I get in over my head in the regulatory space, I'm going to turn it over to my colleague Irene, who's the pro here. So.
Hey, Chris, I think all your regulatory comments hit it right, hit the nail on the head, right? Very well stated and very accurate. We are all learning in this space, including the regulatory authorities, right? And so because of that, we have seen a shift over years in their thinking and the regulatory framework they're putting around these assays. And so from our own experiences previously, A lot of these assays prior to, when taken into clinical trial, we would submit our study risk determinations to the agency, and they would come out non-significant risk. So the FDA didn't need to see the performance of the assays prior to clinical trial. They felt that the safety aspect was not as big of an issue back then. And that has very much changed. So currently, almost every protocol that we bring to the agency that uses an AAV-based assay or a therapy is considered significant risk. And with that come all the regulatory requirements, right? The FDA would like to see, or the CDRH side FDA would like to see an IDE and where they can assess the performance of the assay with respect to safety prior to the clinical trial. And so what that requires on our side is proper planning. How do we de-risk these? How do we give the FDA the safety and assurance they need in these diagnostics in order to begin the clinical trial? And we do that with a lot of communication and very close collaboration with the agency. At the early stages, we as LabCorp try to go in and communicate with the FDA, even prior to development, with the pre-submission, using the pre-submission process. That will allow us to understand, and I think after all the pre-submissions we have done at this point, we're pretty much experts in it, and the communications the feedback does become less and less, and we're finding agreements reached easier on what is needed and what kind of tests they'd like to see for the clinical trial. And so once we put this in place, we do submit our IDEs, and we've been highly successful in getting those approved with the first review. Now, in the EU, that process is a little bit more complicated. We do not have a pre-submission process in place with the competent authorities, which, as many of you here already know, we do need to interact with them through the performance study application process prior to starting a clinical study where a diagnostic is used for patient treatment, enrollment, and monitoring. And so in this instance, what we need to do is just plan our timelines accordingly and expect feedback during that review process. And so we like to give a 6 to 8 month timeline at least prior to clinical study start, in which we submit this PSA. And currently, unfortunately, because UTMed is not at play yet for us with respect to this component, we do have to submit individually. So those timelines have to take into consideration that you are talking to individual health authorities, and then you'll have to respond to each of them individually as well. And so it's planning at these stages. And with each communication, we learn more about what the regulatory authorities in the US and the rest of the world are requiring.
Thank you, Irene, for that added color from a regulatory perspective. Seems very complex just listening to the regulatory considerations that might be involved, but also from a scientific and technical aspect that Chris and Taylor, you touched upon. So I'm going to focus my attention again back to what Chris, you and Taylor just touched upon, which is around the different types of assessments that could be conducted from a potential CDX perspective. Could you elaborate on the different types of assays, assay platforms, or methodologies that might be involved in the development of these companion diagnostics to ensure that the right patient gets that right therapeutic? And again, from that lens of a cell therapy versus gene therapy, because the assessments might vary differently across the two. So Taylor, your thoughts from a cell therapy perspective first?
Yeah, no, a great question and great topic. And I think that's mainly because if you look at cell therapy specifically, as I mentioned earlier, there's no current companion diagnostics. So there is no absolute gold standard that you can point to as that's the technology of choice, right? But there are a number of different technologies that are currently being utilized to assess efficacy, assess potential safety even today. So just by the way of an example that we've been involved with in collaboration with physician scientists at the University of California, San Diego. But in that particular study, we measured persistence of both circulating cell-free tumor DNA, so the liquid biopsy that you may have heard about, with in combination with the viral construct itself. And we were able to link the persistence of those two biomarkers to patient outcomes. Very small cohort, very much a proof of concept, but I think it sheds a little bit of light around some of the technologies that may be able to be used. So in that particular study, we used both a low-pass genome-wide next-generation sequencing assay to measure circulating tumor DNA, as well as droplet digital PCR to have a high-sensitivity assay for measuring the construct. And so that's one particular instance. Another example that you can point to would be around HLA type, right? Especially as we move away from autologous construct. So, for example, HLA is often measured using either long or short read next-gen sequencing, either alone or in combination. And I mentioned that could be important to those cell therapies, where that MHC presentation is really important, like CAR T therapies, for example. So, in addition to that, since they are designed specifically to look for biomarkers that are present on the tumor cells themselves, you can really think about a variety of different molecular techniques that may be able to be used to measure the presence of that particular biomarker. So something like IHC or something like flow cytometry for protein-based biomarkers, or even something like RNA sequencing or qPCR, other techniques to measure transcript levels. So the last thing that I would add specific to the cell therapies, in particular those in cancer, of course, is that as we look forward towards becoming more of a personalized medicine, really utilizing and trying to target that individual patient's individual tumor, you could see how something like comprehensive genomic profiling of the tumor could come into play in order to enable a really personalized approach to that. And so what I would say is, while we haven't seen a companion diagnostic in that space yet, I do think it's likely that we will ultimately see one in the future for cell therapies. But that's something that's already been implemented, as Akanksha you mentioned, as Chris you mentioned, in the gene therapy space. So Chris, maybe you want to go in a little bit more depth about some of the technologies you've been seeing in the gene therapy space.
Sure. And thanks, Taylor. I think at this stage of the game, I'll start out by saying that all methodologies and all technologies are on the table, right? So whatever you really need, it's early enough. There's no standards. We're beginning to see favorites for sure. And I talked about AAV gene delivery and the need for these antibody screening methods. I think one thing to keep in mind is when we're trying to figure out what platform to use and what marker to look at, that you need to go down a checklist to understand the disease that you're trying to address, and that could be severity of disease or prognosis. So, is the therapy designed to improve quality of life, or is it life-saving? Because your risk-benefit may be significantly different, and the types of assays that you need to assess that risk-benefit and treatment efficacy could be different. The patient population, so are you looking at infants? Are you looking at adolescents, adults, seniors? Are there existing and safe alternative therapies that are available? And so how does that impact overall risk? And then the dose, the route of administration, the target cell or the target tissue, and then the genetic payload in the case of AAV. Are you replacing a gene, or are you trying to introduce a system that could repair a defective gene? So a lot of these things come into play when you're trying to assess what type of assays you need to support your gene therapy. I'll just start by contrasting a couple of different examples of applications, and maybe you'll see that need a little better. So in the case of blood clotting disorders or lysosomal storage diseases, there are safe and effective alternative therapies available. So there are enzyme replacement therapies or blood clotting factor replacement therapy. So you need to assess the risk and then accommodate the assay to address that risk. In the case of a severe affliction like spinal muscular atrophy, the condition is certainly life-threatening. There's no alternative therapies. The patient population that you're treating is very young. and may be less likely to have been exposed or have a history of AAV infection. So maybe that reliance on a CDX is a little bit different there. In the case of retinopathies, a lot of times the therapeutic delivery is directly to the eye, so intraocular injections, and maybe neutralizing antibody responses in those cases are really not playing into treatment efficacy. So there could be a lot of differences. I think in addition to, I've been focusing here on AAV gene delivery, but there are other important types of assays that could come into play. So certainly, as Taylor mentioned, gene sequencing or perhaps even PCR assays. And so there are some cases where the defective gene expression that needs to be addressed is due to aberrant splicing of the gene. And so you may need to assess whether or not someone that is a subject of a disease is a candidate for therapy based on what is causing the disease itself. So is it a deletion? Is it a splicing variation? So there are subtleties in even patient population in that regard. And then also some assays are sequencing assays are being used to confirm the disease diagnosis, like I mentioned spinal muscular atrophy. So sequencing of the SMN1 and SMN2 genes to really confirm that is the true cause of the disease. More recently, we're starting to see other types of assays come into play, anti-drug antibody assays, and in this case, I think a good example is in lysosomal storage disease, where for some time now there have been enzyme replacement therapies. If you're trying to or if you're interested in converting someone from an enzyme replacement approach of treatment to a gene therapy approach, it may be important to understand whether over the course of enzyme replacement therapy, there has been an opportunity to develop anti-drug antibodies. And maybe, in the early days of clinical trials, you avoid anti-drug antibodies. So again, everything's in play here. It's early days. And I think with that, I'll just close. And Akanksha, maybe we can move on to the next question, or maybe you want to have some comments about my comments and Taylor's comments. Oh, excuse me. Should have silenced my phone.
Well, no, that was very insightful, Chris and Taylor. Both of you alluded to the complexities that might be involved, whether it is at the assay level, the type of platform, the patient population, the indication, right? It's definitely a complex factors that are in play here. And then from a perspective of a companion diagnostic development, there's that need to align the development timelines with your therapeutic development, right? So are there any steps that may be recommended from the perspective of a companion diagnostic development and commercialization strategy, right? Any insights that you all can share with our sponsors on how to best strategize, plan, and prepare? I heard Irene earlier, right? Plan. How do you plan? How do you prepare so that you have a successful companion diagnostic available at the time of launch of your therapeutic? You have your strategy in place. You have your commercialization strategy in place. Any thoughts that you guys can share? And I'll look at Chris. Would you like to take a stab at this first?
Sure. I'll give it a first shot. So I agree. I love Irene's comment of plan, and I think it should be followed by execute. So, but my best advice, I think, based on this experience so far, is that you really want to engage, a partnership early. It's probably never too soon to start talking that for the sponsors, the therapeutic sponsors, and the diagnostic developers to start talking. There's just a lot of activities that require this planning that Irene mentioned. So there's the development itself. Maybe the assay needs, de novo design. Maybe you're not quite sure what assay you need to develop or how you're going to do it. Then there's the development itself, followed by the validation. You have to move into manufacturing, implementation. Even after the commercial launch, you have to worry about post-launch monitoring and for safety and efficacy. So, it really, it really should be a partnership. One shouldn't think about, services offered and then moving on. So I think for LabCorp, the most attractive model is to develop long-term partners to support each other's programs. Even in the cases where there might be a prototype assay that already exists, and that assay needs to be transferred to a lab that can take it through the companion diagnostic process. It's never going to be a straight transfer. There's going to be assay rework. There's going to be bridging studies that are needed to bridge to the earlier clinical data. So one has to keep that in mind. You can't just run off at the last minute and say, I have an assay, it's ready to go, because the reality is it's probably not. And I'll just go back to that earlier comment that I made, that there are no off-the-shelf solutions at this time. There just aren't. I will come back to regulatory, and maybe Irene can comment, or maybe Taylor should comment first on cell therapy. But LDTs in general are falling out of favor with the regulatory agencies. So it really, back to Irene's point, you need to engage early and consider the regulatory strategy.
Yeah, no, thanks, Chris. And I'll say a couple of words, and then absolutely, I think everything gets tied together by all the regulatory components. So I'd love to hear Irene's thoughts as well. But I mean, Akanksha, you mentioned it earlier, right? If you think about what is a companion diagnostic, it's a test that's designed, developed, validated, and then ultimately commercialized for one primary purpose. It's to ensure that the right patients, those that are the most likely to get, to benefit from that drug, are ultimately receiving that drug. So how do you do that? And so if you think about it, it really comes down to, answering a fundamental but often complex question of what's the best way to do that? What's the best way for that testing to occur so that you can identify the right way for that particular patient population to receive that test so that they can receive that drug? So the first thing that I would recommend, and it builds on some of the stuff that Chris mentioned as well, is you need to first understand the breadth of the patient population that needs to be tested, right? So this often linked to the prevalence of the disease or the local or global population distribution. So as an example, Chris mentioned earlier, if you think about a case like a gene therapy targeting a very rare disease, or something like hemophilia B, like that model may be best suited by a single site or a limited site manufacturing and testing model, where samples may be distributed to, like I mentioned, either a single site for testing or a limited number of sites, because the incidence rates in, if you distribute that broadly, may not warrant all of the work that's required to implement and offer that type of testing. But on the other hand, the disease is more common, like, for example, some diseases that are currently being evaluated, like heart disease, for example, and looking at gene therapy options there. A different model may be the right approach, right? One that utilizes a large-scale testing and widespread, potentially global distribution. Maybe that's the best model in that cohort. So I really think it's critically important to understand the disease, the severity of the disease, what that turnaround time needs to be, as Chris alluded to, but also understanding the overall demographics of what may be best suited, or what's the best approach to enable that testing to reach as many of the patients that could benefit from it as possible, right? And all of these different, development and manufacturing considerations need to be tied together by a robust underlying regulatory framework. So Irene, I don't know if you want to comment on how those things can be brought together from a regulatory perspective.
Yeah, absolutely. So I think commercialization from the regulatory side, right? I like to take a step back and think of, well, what's my biggest hurdle towards commercialization? What do I need to do? What do I need to make sure is in place? And the obvious answer is a successful clinical trial, right? You need to be able to show the safety and efficacy of your drug. So from the device side, what needs to be in place now to ensure that that successful clinical trial happens? And it's really about selecting the right patient population. And how do you do that? It's about having the appropriate cut points. And this is also a question that we see the FDA asking more and more, and they are asking for your reasoning behind that cut point. So from early on at the developmental stage, we work with our pharmaceutical partners to really determine what that cut point is based on the data that they have, and ensure that we are selecting the right patient population for that clinical trial. And it's, when the FDA asks us this question, it's not that there's a right or wrong answer or that they have a cut point in mind for you. It's more to prepare you and to let you know that we need a reason for this. How did you select it? So how do we know that you are truly picking out the appropriate patients and we're not putting anyone at risk? The FDA's biggest concern around these programs right now, as it seems, is treating a patient that is a false negative. So false negative meaning they don't have the AAV antibodies, right? And they do have AAV antibodies and they are treated inappropriately. And so what happens with the adverse events associated with that and long-term mutagenesis is a point that they have also brought up. So showing accuracy on that cut point is critical. Also to take under consideration early on in your development is your commercialization plan. So as Taylor, as you suggested, global distribution and assay or a single site, it makes a difference in the requirements as far as maybe some of the studies that would be required, and also how you will be testing these samples later on. Do you need a globally distributed kit, or will samples from rest of the world be coming to a single site in the US or anywhere outside of that particular country? And that makes a big difference because regulatory requirements are very much linked, globally regulatory requirements are very much linked to the specific country and that model. There are a number of countries that do not regulate testing of samples outside of their own country, for example. So as a single site in the US, it would alleviate some of the formal submissions and regulatory burden in certain countries to have the sample shipped there. You might not need a formal submission. In other countries, on the other hand, they may have a different process in place that would allow them to approve this assay that is not in their country. In Japan, for example, we have the example of they want a software that the physicians there can interact with to be the medical device that gets approved. And under that pathway, the assay is also reviewed. And so that sort of due diligence and planning, again, takes time. And so to know the countries that you're going to go into from the beginning or that you're considering will really help the regulatory team lay out the appropriate structure for development and also what needs to be put in place from a testing perspective and documentation wise. And of course, after approval, there's considerations around test availability and the payer components. And Taylor, I know you have a lot of thoughts in that space, so I'm going to pass it over to you to tell us a little bit more about.
Thanks, Irene. Yeah, and I mean, we touched on a number of different topics. We talked about development, we talked about manufacturing, the underlying regulatory landscape and the requirements or best practices there. But I think it should at least be noted that both drug developers and device manufacturers alike should ultimately consider the ultimate reimbursement model for both the CDX, but also the therapy, and make sure that they're aligned in that regard to truly identify the optimal approach in that for each individual program, because it can certainly vary. So, for example, there are some instances where the cost of the testing is covered by the pharmaceutical company themselves, the drug developer. And that process can often accelerate the availability and utilization of companion diagnostics because it can eliminate some of the more long-term requirements of work to interact with varying reimbursement bodies, whether those be private payers or government-based reimbursement structures like we see in various countries. But in other cases, that approach, it may not make sense for a number of reasons. And a more, I would say, quote unquote, traditional reimbursement path may be the one that makes the most sense. So I think something that's worth noting in that regard is that the requirements often differ significantly from region to region, from country to country. So it adds significant complexity to some of that commercial planning. So, I think all of those things can be worked out, can be identified the best approach with, a consistent amount of work. But I do think in the end, it's very important that the sponsors, the drug manufacturers, and the test manufacturers, device manufacturers, align early on in that process to determine the best path for commercialization to ultimately yield what everyone wants, right? Which is to have a successful therapy that is supported by the associated companion diagnostic.
Thank you all. This was very, very insightful and very complex, right? Complexity from an assay perspective, cutoffs, regulatory, commercialization, payer, market access, and then the three-way partnerships. Definitely a lot to consider. So Irene, if I may, I want to pick your brains a little bit more from a regulatory perspective, right? You mentioned, right, it's complex, it's evolving, sometimes often not clear. The process might be different based on the geographies that you're trying to target, right? So any insights or perspective that you can share as to how best you can strategize, plan, and prepare to navigate that regulatory process that's so complex? And again, it might be different, from a geography perspective, right? US versus EU or other parts of the world. Any perspective and thoughts that you could share?
Absolutely. So I think we discussed a little bit, and I feel like I broke a word about planning, right? And in the US, a lot of it happens at the developmental stage, and then you get into the clinical trial, and it becomes a more familiar process. You go through the trial, it's successful, the CDX and drug manufacturers submit their respective submissions in parallel. You interact with the FDA during the review process, and hopefully all goes well, and you receive concurrent approval, right? So at the same time, the drug and device are expected to be approved. And the next day they can be used commercially in the market. Now in the EU, where the process is newer to industry as well as to the regulatory authority there, that's where additional planning, like I said before, is key. And I feel that's the regulatory mind. We plan and we strategize. We plan and we strategize. So I apologize for the repetition. But at the initial stages, the diagnostic manufacturer must select the notified body to review their technical file. And we do this years prior to the expected approval. So at least 2 years ahead, we will go in, we'll connect with we'll find out that we have, as LabCorp, we have our notified body that we work with, and we will inform them of this pending submission. If a facility doesn't have ISO 13485 certification, of course our CDX sites do, then you would need to work with them as well for that certification. The contracting process alone takes several months, which is why we start ahead. And then once that process and contracting is in place, we align in a submission date that could be months out. They are very, very busy as well. And they have an influx of submissions now with IVDR and all the requirements. And so they are also learning. And so their resources are a little constrained. And it's taking them time to review the submissions because it might be the first one of its kind that they've reviewed. And so once we have a date in place that we expect to submit, from then on, we budget about a 2-year timeline from receipt of the submission to final CE IVDR marking. And so this is where planning, again, right, is key. Because what it looks like right now, whereas in the US, there is that parallel review, and the expectation is that CDRH and either CDER or CBER will have that approval on the same day. That sort of framework does not exist yet in the EU. So your notified body takes as long as they need. In order to have their review complete, we've been informed by our notified body that they'd like to do it under a year. That's their preference. So we assume about a year. Then there is a coordination process with EMA. It takes them about 3 months to send the material and have EMA prepare in order to receive the documentation. Another 120 days upon which EMA reviews, asks questions. That doesn't include the stop clock. That is their time. And then finally, upon positive opinion, we can expect the IVDRC marking, right? So it's about a 2-year process. And there's a lot of questions around that. The drug timeline, for example, for approval might be a lot less. And what happens in that case? And unfortunately, those answers are not in place yet. We've asked all that, what happens if the drug is approved first? Can they use an available assay on the market that is not CE marked in the timeframe to make, to not leave patients without treatment? And the answers are just unclear right now. It doesn't seem like the regulatory or the notified bodies themselves have those answers. As LabCorp, of course, we are working through various organizations to voice those concerns, and we hope to see some movement in that area. But as it stands right now, there are no definite answers. And so all we can do is get in on the process early and hope we win enough time on the CDX side to get as close to that drug approval as possible.
Thanks, Irene, for that color from a regulatory perspective. So from what I've heard so far from all of you, right? Cell and gene therapy, very exciting, innovative field. There's a play here for companion diagnostics, ensuring that the right patient gets that right therapeutic. As you look at this arena, are there any challenges or limitations that you see from a perspective of companion diagnostic development for cell and gene therapies, and how the industry might be looking to address some of those? And Chris, I'm gonna direct this question to you first, and then also ask our panelists to weigh in for this discussion as well.
Sure, thanks, Akanksha. I'll start with limitations. I think I touched on this very early on, that for AAV-directed gene therapy, 50% of the patient population may not be a candidate for treatment. So that's a big challenge. And so there are efforts now in many laboratories to try and develop methods that would enable the treatment of the AAV neutralizing antibody positive patient population. And that could be by various immunosuppressive methods prior to dosing, or actually physical methods where you perhaps pre-treat the patient with enzymes that degrade antibodies prior to dosing. So there's a lot of work and interest going on there to try and understand how to treat that patient population that's really not eligible at this time. With respect to lentiviral vectors or retrovirus vectors, I think one of the limitations is the risk of adverse events that are due to inappropriate integration of the virus, or I wouldn't say inappropriate, but integration of the virus through its normal life cycle into sites of the host genome that could disrupt important the expression of important genes, for example, tumor suppressor genes, or that would get knocked out, or perhaps the upregulation of oncogenes that could cause the formation of new tumors. So and, those are all challenges with the available methods of gene delivery, and maybe some non-viral gene methods or delivery methods are the answer to a lot of this. The other point that I'll bring up is that as partners, the therapeutic sponsor and the diagnostic company have to figure out how they're going to support the production of critical reagents that go into these assays. So let's go to AAV for an example. The AAV is used to produce a therapeutic. That therapeutic itself could be used for total antibody binding assays, but for AAV cell-based assays, you need to engineer a reporter virus, something that, for example, that has a reporter like luciferase in it, and is then encapsulated by the same capsid that's used for the therapeutic, to make sure that you're measuring the very specific antibody responses to that capsid. Some of these capsids are available commercially. I think many of you are familiar with them, and they include AAV5 and 6 and 8 and 9, maybe that solves the problem for those. But there are other capsids that are less well distributed and maybe proprietary. And so how do you create these reporter viruses with proprietary capsids? And the FDA also is very particular at this point about the positive control and its specificity for the capsid. So the capsid has to, the antibody has to match the capsid down to the point that it should be a monoclonal antibody that's directed, that is actually generated through the immunization or of the specific capsid. And so who is going to produce that monoclonal, who's going to manufacture it. So it reminds me, and actually what triggered this thought was Irene's comment about Japan and the special needs to bring the device to Japan. So what third-party relationships are needed for the manufacture of and maintenance of critical reagents in the assay. And the final point I'll make is goes back to the assays themselves and their cutoffs. And so we know that there is a concern at this time for false negatives, as Irene spoke, and that would be dosing individuals that are inappropriately dosed. But on the flip side, there are false positives, and false positives in this case would lead to the failure of an opportunity to treat a patient that could benefit from gene therapy. And this all comes down to how well the assay performs around the assay cutoff, and what are those cutoffs. So I think what keeps me up at night the most is what are the cutoffs based on very limited clinical data. I mean, you don't have a lot of clinical data to go on, and how do you set those cutoffs? And how do you understand the variation that you see around those assay cutoffs? Because the variation in the assay itself is not linear, and so you really need to understand the cutoff before you can describe the variation around the cutoff to the regulatory agencies that take that into consideration. And I'll just close by saying that every gene therapy or cell therapy is different. And in the case of cell therapy, the therapy for each and every patient is different. And so, it's very complex at this time. And Akanksha, I think I'll turn it over to my colleagues to embellish more.
No, I think you touched on, probably the biggest challenge in the field in general for cell therapies, Chris, right? Like, how do you solve the manufacturing requirements? What are the right practices? How do you do that? That's probably a topic for a whole different discussion that could go for another hour. So I think we'll leave that as is. So I'll just touch on a couple real quick on the cell therapy side. And I think one is what is the optimal biomarker and the immune cell type, right? So if you look just at AML as a singular example, there are dozens of different tumor antigen and immune cell type combinations that are currently in trials. So there's still a lot of learning to do and data to be generated in this relatively young field. And then I guess the other one that I would kind of point to specifically is the vast majority of cell therapies has some degree of initial efficacy. Upwards of 80-90% can have some initial efficacy, but many of those responses are short-lived. So I think it will be important to continue to determine how to best or better prolong this initial therapeutic efficacy. So maybe there's something specific to that individual patient or to the cells where the use of a specific biomarker and that could lead to a potential companion diagnostic for long-term therapeutic outcome should be studied and hopefully elucidated as we move forward. But I think to summarize this field, it will need to continue to rapidly evolve through that new research, through that clinical trial data, importantly, Chris, as you mentioned. And I think it's important for the drug developers and the device manufacturers to work together to ultimately, reach the goal of improving patient outcomes. And Irene, I know you touched on a number of different regulatory challenges, especially with the EU a few minutes ago. Is there any, short follow-ups that you would like to include there?
Oh, I think you have both voiced my concerns very well, right? To the points around cut point and accuracy. I think it's the regulatory concerns are covered.
So thank you all for that very insightful discussion. In the interest of time, let's look at some questions that have come in from our audience. And the first one is, what are the requirements for clinical laboratories to be certified and accredited to perform companion diagnostic testing? Irene, I think this falls more into your realm of expertise. Do you want to take this one?
Of course. Yeah, of course. So in the US, CLIA certification is required for facilities that are performing tests on human specimens for the purpose of diagnosis, prevention, or treatment of disease, or the assessment of health of a human being. It is required in both commercial and clinical testing. Also required for clinical testing, of course, is compliance to GCPs, an international ethical standard for international ethical and scientific quality standard for design, recording, and reporting of clinical trials. Globally, I would say obviously ISO 15189 is a recognized accreditation that would apply to medical labs. Of course, I would expect globally also that medical labs follow and comply to the requirements of their local regulatory bodies, with the understanding that there might be some overlap between those and the ISO 15189 standard. Now, if you're talking about retrospective or exploratory studies where data is not provided for diagnosis or treatment, in those instances, a CLIA lab would not necessarily be required, and RUO testing may be conducted. But ultimately, the rigor you want to put around that also depends on what you're going to do with those results at the end, and the sort of documentation that you want in place. If you think there's any opportunity for something to become registrational afterwards.
Thanks, Irene. There's another question, and I think this is for you, Chris. Can you elaborate on the approaches that are used to establish the cut point for anti-AAV assays if the asset has never been in humans?
Yeah, boy, that's a tough one. There are cases where the asset has been in humans and there is some phase 1, phase 2 data. Oftentimes those numbers of patients who have been dosed is tiny, but you may get a glimmer. I think at this point, the default is to be fairly conservative. So really look at assays that are very sensitive for the detection of neutralizing antibody and begin there. And then based on the data that one sees in trials, then consider relaxing the assay cut point. And again, this may depend on the therapeutic and the indication and the mode of delivery, a lot of other factors. But I can't say that there's a formula to use, unfortunately.
Thank you, Chris. I think in the interest of time, let's take some of these questions offline and answer. I would really like to thank the audience for joining the call today and listening to us and their questions. And then also to our panelists for providing their insights. This was indeed very helpful. So thank you all.
All right. Well, thank you, Akanksha, and thank you to our panel of speakers for your time today and your important research. We would also like to thank LabRoots and our sponsor, LabCorp, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.